Hello guys, good day. And in today's topic or lecture, I'll be talking about genetics. Genetics genetics deals with lots of things relating to the reproduction of human beings. It deals with the genetic factors of human beings, how you got your gene, how or why you are a female, how you became tall, how you became dark or fair. So most people find genetics really difficult to understand, but if you really understand the basis of genetics, it becomes really easy and simple for you to understand genetics. So as we go, when we talk about genetics, we'll be having some course outline. The course outline for this topic is common genetic terms, heredity, sickle cell anemia, and then genetics of blood. So by the time we finish with this slide, we'll be able to know the common genetic terms, heredity, sickle cell anemia, and the genetics of blood. My objective here, or our objective here, is to understand the concept of genetics. And then so when we say genetics is a branch of biology that deals with the study of genes, genetic variation, and heredity in organisms. Genetics can also be defined as the study of genes and tries to explain what they are and how they work. So basically, genetics is the study of genes, how they come about and how they combine or function together to form a human being. So with the common terms, we have chromosomes. Chromosomes, these are thread-like structures present in the nucleus of an atom of a cell. Like every cell has a chromosome because it determines the character of the individual. The chromosome has the DNA and the genes on it. So an individual with a chromosome is specified by it. Everybody is special to what he or she is. We have even twins were born by the same womb and went through the same processing, but they are still variant due to the types or the number of chromosomes or like how their chromosomes function. And then we'll talk about genes. Genes are hereditary particles located in the chromosome. Genes are how living organisms inherit features or traits from their ancestors. For instance, look at the way you look. You look the way you look because you inherited your parents' features. So like when we talk about your features, you are a male, you're a female, and you are dark, you have black hair, you are short. All these features were features that you inherited from your parents. It's not something you took from somewhere else, but from your parents, because you took each gene from your parents. So like all these features you see on yourself is something you inherited from your parents. If you see it in them, then it's their phenotype. That means you inherited it straightforward. But when you don't see it, sometimes they are recessive. So you took it and then you became dominant of it. When we go on, as we go on, we get to understand all the terms. When you understand the terms in genetics, it becomes really easy for you to understand the concepts of genetics. So when we talk about traits, there are major and minor features inherited by an organism. For example, facial dimple, eye color, and then a chinklet. Chinklet is like that Y shape kind of thing on the chin, like when it's mostly found on ladies. When ladies smile, you see that kind of Y shape on the chin. Sometimes it looks really nice. So as we go on, the diagram you see here is an illustration of gene and chromosome. The chromosome is seen right here, and then the gene is seen here. Then when we come to zygote is a single cell formed as a result of a union of a male sex cell and a female sex cell during reproduction. So at the end of the fusion of the sex cells from each parent, they form a zygote. Okay, so we go on and then we talk about alleles. They are the alternative form of genes occupying the same place of chromosome and affecting the same character but in different ways. Like for instance, let's say maybe a sickle cell, but it's recessive. So you are, let's say AS, A and a small s, meaning you have one A, which means you are normal, and you have one small s, which means you are a carrier. So when you look at the example, it's A or small a or big A. You may have both, and it shows that the small a is recessive of the big A. And we go on to look at homozygotes, a condition when an organism contains identical alleles, and then the heterozygotes, a condition when an organism has two different alleles. So like in this example, the homozygotes, it's homo means same. So like assuming you have sickle cell, so if you have SS, it means you are homozygous to sickle cell. But when you have A and a small s, it means you are heterozygous to sickle cell because you have a smaller portion, the carrier portion of the sickle cell. So as we go on, phenotype, this is an outward appearance of an organism. When we talk about the phenotype, mostly in genetics, most individuals look at their outer of their phenotypical appearance and then they judge themselves. You can have an eye color, but you can have an eye color which is green, but your phenotype is showing brown. Mostly, it won't have an effect unless you are crossed with an individual who is also having a recessive green. Then you realize that your kid is going to have a green eyes. So even though you think you are short, but if you are to go into your genotypical perspective, you realize that you may be tall, but the phenotype overcame the genotype, so you became short. So it doesn't mean that because you are short and then you try to look down to yourself. No. When you want to look at the genotype, it is a sum of genes inherited from both parents. For instance, the genes you inherit from parents, like the height, your sex, whether you're female or a male, and all those things are things you inherited from your parents' chromosomes. So the genotype is basically the sum of the genes you inherited. And the phenotype is the appearance, how you appear, your height, your skin color, your hair color, how you appear to the outside world is the phenotypical expression. So when we talk about dominant character, this is a character that shows its effect in the presence of a contrasting character. So we were talking about it earlier. The dominant character here, it's like a superior character. This character overshadows all other characters. Like assuming I've been using sickle cell a lot because it's easy to understand with sickle cell. Assuming you are a sickle cell, but you're a carrier. So like you have AS. This A is dominant over the small s. This would make you not to be a sickler. But assuming you were having SS, that one the S is dominant. So the S is showing its characters or its effects on you. That's when you become a sickler. When we talk about the recessive character, they don't show their effects, as in the A and the S, even though the sickle is there, but still it's not showing effect in an individual who is having AS. So that's basically how it's like. And then when we talk about F1 generation, the generation produced by crossing homozygous parent stock. So like when you take two parents and then you cross them, the generation you get is the F1 generation. And then with the F2 generation, when you pick two of the F1 generations and then you cross them, the generations you get after that is so called the F2 generation. Okay, so we go on to heredity. Heredity, the transmission of characters from parents to their offspring. During fertilization, traits are transferred to the offspring. Inheritance is the transmission of traits to offspring. So basically, heredity is just you getting your characters or you having your genes from your parents. According to Roman, the law of segregation of genes states that the characteristics of an organism are determined by the internal factors called genes. According to Mendel's first law, he was saying that the characteristics of an organism is determined by the internal factors called genes. Even though like you may have in your genes to be white in color, but your phenotypical expression is dark or a chocolate color. According to Mendel, to determine the characteristics of an organism, you must check the internal factors, which is the gene. Genes occur in pair, but only a single gene can be represented in a single gamete. So like the genes, they occur in pair, but only one shows its character, unless one is recessive and then the other is dominant. We'll talk about monohybrid inheritance in humans. So when we come to the topic of albinism, it's a condition in which the skin pigment fails to develop and results in having a light skin, white skin, or pink eyes. Albinism is caused by recessive gene A on the autosome. Autosome is humans, we have 23 chromosomes. The 23rd chromosome determines your sex, whether you're a female or you're a male. The 22 chromosomes left are called autosomes. So albinism is caused by recessive gene A on your 22 chromosomes. So the dominant gene for a normal skin color is A, and the genotype for a normal skin person is AA or A small a. Here, the A small a, the big A is dominant over the small a, meaning even though you have a career of being an albino, but it's not showing you. So it means that your phenotypical expression will be normal. But when you go to your genotype and you want to know your characteristics, you will realize that you're a carrier of sickle cell. And then the genotype for albino skin is small a. That means this individual is going to be affected fully by albinism. And then at the end, the person is going to have light skin, white hair, even though the person is not old, but still going to get white hair, pinky eyes, and then a lot of issues. So in these illustrations, we have a snake. We have a snake who is an albino. We have a child. You can see the hair, you can see the face. Realize that it's being affected by it's not a career. This part, this the albino is dominant over the normal albino, the normal color. That means this child is not only a carrier but a holder. So the child is AA, the snake too is AA. When you look at the snake, you see that the color is fading, like the snake is, even though it's having white color, but it's white and pale. So this is an example of an albino human being and an animal. In the other diagram, this shows a carrier father mating with a carrier mother. A carrier mother having albino and then a carrier father having albino. Even though they aren't affected by the albino, but they had their carriers in their genotype. The possibility of they giving birth in their F1 generation, they may give birth to a child who is not affected. They may give birth to a child, two kids, a percentage who are carriers of the albinism, and then they may also give birth to a child who is affected and is a carrier, and it's not only a carrier, and is affected by the albino. So you realize that the the ratio will be 1:2:1. So when we want to avoid albinism, the best way to avoid it is that people who are carriers shouldn't get married or give birth, because when they do that, there's a possibility they'll give birth to a child who is an albino. And then a carrier father shouldn't also marry a sickle cell, because there's an albino, because there's a possibility that they are going to give birth to an affected child. Okay, so we go on. So we'll talk about back crossing. A back crossing is a cross between an offspring in the F2 generation and a recessive parent. The genotype of each offspring in the F2 generation can be determined with back crossing. So we want to determine the genotype of an offspring in F2 generation. We just cross the F2 generation and the recessive parent to get, so the generation you get will be, that generation you get will determine the genotype of the offspring. For example, capital T, that means tall, and then short. And then in the example, you realize that a black and then albino, they are matched, and then we get AB. And then a black and albino are matched, and we get AB. So this individual, the ones that are like the F2 generations, being crossed by the recessive parent. So we'll talk about a test cross. Test cross is used to identify whether the phenotype of an organism showing a dominant trait is due to homozygous or heterozygous genetic conditions. To identify whether an organism is exhibiting a dominant trait as homozygous or heterozygous, for specifically, as a scientist can perform a test cross. The organism in question is crossed with an organism that is homozygous for a recessive trait, and the offspring of the test cross are examined. So here, an organism in question, that organism you want to find out whether the dominant trait is due to the homozygous or heterozygous genetic condition. You cross that organism with an organism that is homozygous for a recessive trait, and then you examine the results you have at the end. So in test crossing, F2 individuals with a phenotype of dominant parent with a recessive parent are crossed. If the F2 individuals in homozygous dominance of the given trait, then all the offspring will definitely be dominant characters. They will display some kind of dominant characters because the F2 individual is homozygous. That means like it's, for example, let's say it's AA, and then it's AA, and then you are crossing it with a heterozygous parent. Let's say another parent who is A, capital A, small a. You realize that all the offspring will be dominant characters. However, if the F2 generation is heterozygous, then the offspring will show a ratio of 1:1. But this is assumed when the F2 individual is heterozygous in the sense of A and then small a. When you cross this with a recessive parent, you realize that the ratio will be 1:1. We may get a dominant offspring, and then we may get a recessive offspring. That means it's 50-50. The possibility of having a child who is dominant or is not affected by a trait is 50, and the possibility of the individual being affected is 50%. So in this exercise, you are required to work it out, and then if you have an answer, you can put it in the comment section or in our group, and then on our WhatsApp group, and then we'll take it from there. independent assortment. So the law of independent assortment says that the alleles of two or more different genes get sorted into gametes immediately of one another, independently of one another, sorry. In other words, the allele a gamete receives for one gene does not influence the allele received for another gene. For instance, the transmission of genes determining the height and seed color are independent events. This means that the gene determining the seed height are located on one pair of chromosomes, and the gene determining the seed color are also located on another distinguished pair of chromosomes. This independent assortment is not any big deal. From the instance, you realize that the height of an individual, the determining height of an individual, is found on a pair of chromosomes, whilst the seed color or the color of an individual is found on another pair of chromosomes. So here they are all brought into action, or they show their effect since they are found on different chromosomes. And then we're go on to dihybrid inheritance. Dihybrid cross describes a mating experiment between two organisms that are identically hybrid for two traits. A hybrid organism is one that is heterozygote. That means for instance, let's give an example. A big A and then a small a. A hybrid organism is having a trait such as a big A and then a small a, which means that it carries two different alleles. So you see here, we have two different alleles at a particular genetic position. So one can be recessive and then the other can be dominant. So in our example, let's look at a pea plant and say the two different traits we are examining are color and height. One dominant allele, H for height, and one recessive, H, small h, which produces a dwarf pea plant. So this is basically the big H and then the small h as we discussed. Then we come to monohybrid and dihybrid. Monohybrid cross is used to study the inheritance of a single pair of allele. But here, in the dihybrid, we use the cross to study the inheritance of two different alleles. In the monohybrid, when you cross, you determine the color of an individual, whether the individual is having is an albino or is not. But with the dihybrid crossing, you realize that we can find out the height and the color of the individual all at the same time. The diagram shows the crossing between AL, where we have A and then A, big M, capital big A, and then the small a. And then we have the capital L and then the small l. Okay, so, and then we have the monohybrid 2 here, crossing and then determining in one single pair of allele. Now we come to linkage. Linkage is a close association of genes or other DNA sequences on the same chromosome. The closer two genes are to each other on the chromosome, the greater the ability they will be inherited. So when two genes are close to each other on the chromosome, they are called to be linked, and they have a greater probability that they will be inherited together. And then we go to sex determination. Most organisms are made up of two sexes, the male and the female sex. For example, in the case of humans, dogs, and some flowering plants. In other organisms, they are bisexual, mostly the case of higher plants and some lower vertebrates. And some plant sex are also determined by the kind of environment they find themselves. Mostly I say this kind of plants are lazy because when the environment is not adequate for growth, the plant develops to a male. And then when the environment is adequate, when the environment is adequate, the plant goes into a female. But when it's not, then the plant grows into a male. So some plant sex are also determined by the kind of environment they find themselves. There are 22 pair of chromosomes in the nucleus of every man. The 23rd of the chromosome is called sex chromosome. We talked about this earlier. It is called sex chromosome or heterosome because it determines the sex of an organism. So to determine the sex of an organism, we check the 23rd chromosome. So the other 22 chromosomes are called autosomes. In humans, there are two sex chromosomes, the X and Y chromosome. The X chromosome is the female sex determining character, whilst the Y chromosome is the male sex determining character. The Y carries few genes since it's short, and the female have two X chromosomes in their gametes, and hence are referred to as homogametic. And then the males are also heterogametic because they have an X and then a Y. Even though the Y chromosome is short, it's really strong or it's effective in its action. And then we have abnormal individuals who may be male but still may have XXY, XXXXY, XXXXXY. These individuals are abnormal. They have abnormal chromosomes, but they are still male. So the probability of giving birth to a male or a female is 50-50. In the case of crossing a homogamete mother and then a heterogamete father, the possibility of giving birth to a female is 50%, and the possibility of giving birth to a male is 50%. So the sex of an individual is determined during fertilization. When the sperm of an individual fertilizes the egg of a female, mostly the sperm that enters that fertile fuses with the egg determines the sex. We have X and we have X sperms and we have Y sperms. So when the Y sperms fuses with the egg, we produce a male. But when the X sperm fuses with the egg, then we'll get a female. Mostly, we say most mothers are blamed for giving birth to only females or males. Mostly, this is the case that the sex of an individual is determined by the type or the kind of sperm that fertilizes the egg. So mostly, the Y sperms are fast and strong, and then the X sperms they are slow. They are slower than the Y sperms. So if you want to give birth to a girl, then if you are a woman and then you ovulate, and then the best time to have sex in order to give birth is to have sex before the egg is released from the fallopian tube. Because so assuming you want to give birth to a girl, you would have to first have sex before an egg is released. This is because the Y sperms swim fast to get the egg. So as time goes on and then they get to the womb they get to the fallopian tube and then they realize there's no egg. The Y sperm fades off and dies off. But since the X sperm is slow, it takes time to reach there. So a sperm can stay in a human for 5 days. So to give birth to a girl, you must make sure that the time the egg gets out of the fallopian tube is delayed so that the Y sperm doesn't get to egg earlier. But if you want to give birth to a male, then the best thing is when you realize you are in ovulation, then you should have sex at that particular moment, because it's possible the Y sperm will get to the egg and fertilize it. So that's a trick you can use to give birth to, even though it's not really scientifically proven, but it's something that when you understand and practice it, it's really going to be of help. And then that's how the X chromosome and then the Y chromosome look like. The Y chromosome is short and then the X chromosome is long. So go on. Then we'll talk about sex linkage. Sex linkage is a trait in which a gene is located on the sex chromosome. In humans, the term generally refers to traits that are influenced by genes on the X chromosome. In a sex-linked disease, it is usually males who are affected because they have a single copy of X chromosome that carries a mutation. For instance, some diseases like um, let's say sickle cell or any kind of disease, the X chromosome carries the mutation. So if the S becomes a carrier, the Y doesn't carry the mutation. So it becomes difficult for you to have a dominant and a recessive character. So here, since the X is having the trait or the disease, it takes it. That's why we say it normally affects males, because in females we have another X chromosome, because it's possible to have a recessive or to have a dominant. So let's say you inherited a sickle cell or any kind of disease from your parents and it's on the X chromosome. Gene mutation. If the other X chromosome is dominant over the disease on the other X chromosome, it is possible you won't get affected. But since in the male they have only one X chromosome which carries out a mutation, it is obvious that they will get the disease easily. And then we come to talking about crossing over. Crossing over is the exchange of genes between two homologous chromosomes. As a result of the chromatic breaking off and rejoining to another partner, it results in the separation of previous linked genes. Genes are linked on the chromosomes. So it gets to a time there's this kind of crossing over. A part of a chromatin breaks off and then joins to another chromosome. The reality behind this is that even though it causes a break in the linked gene, but the crossing over is a combination of new genes, hence it results in genetic variations in organism. It doesn't mean that it's bad that those genetic factors were cut off and the new ones came. Sometimes it's good to cause variations in organisms, and since variations should be caused in organisms, these things occur at the genotypic level. So the chromatin will break off from a part of the chromosome and then join another part, and then a new chromatic part will join the other part, forming a whole individual with different genetic factors, even though they got the genetic factors from the same parent. And I'm going on to lethal genes. Lethal genes are genes that cause the death of the organism that carries them. They are usually a result of mutation in genes that are essential for growth or development. Lethal alleles may be recessive, dominant, or conditional depending on the gene or genes involved. Example of lethal gene is sickle cell anemia. So sickle cell anemia is inherited from parents. It can be recessive, it can be dominant, depending on the gene or the genes involved. So we come to incomplete dominance. Incomplete dominance is when a dominant allele or form of a gene does not completely match the effect of a recessive allele, and the organism resulting physical appearance shows a blending of both alleles. It is also called semi-dominance or partial dominance. Like for instance, instead of the red being dominant over the white, we don't see any dominant and recessive. They are all showing their effect. Here we call it incomplete dominance. Here you realize that the flower here is having red and white and is now having a pink color. You realize that it inherits both colors and then it shows them. Here you don't see the dominant, which color is dominant over which color. So when we talk about here in incomplete dominance, it means you don't see the dominance of the two genes. That means if the character is red, or let's say the character is red and white, you won't be able to determine whether the offspring inherited the white gene or inherited the red gene. All you know is that the generation inherited both without having a dominant or having a recessive. Like when it comes to talking about sickle cell anemia. Sickle cell anemia is an illness in relation to the inheritance of sickle cell traits from one's parents. In sickle cell anemia, the normal hemoglobin in the red blood cell is entirely replaced by abnormal hemoglobin known as hemoglobin S. Sicklers are mostly resistant to malaria parasites since they are not able to attack the red blood cell. Sickle cell anemia is an illness you don't get from an environment. It's something you get as a result of a trait you inherited. For instance, if your parents are carriers of sickle cell and then they are cursed, there's a possibility they'll give birth to a child who is a sickle cell anemia. So sickle cell anemia affects the hemoglobin in the red blood cell. It affects it in the sense that it changes the shape of the cell, and then it affects the function of the hemoglobin. Instead of absorbing oxygen and transporting it to the all parts of the body, it becomes difficult for the body, the hemoglobin, to perform this function because it is replaced by an abnormal hemoglobin S. In the case of malaria parasites affect the red blood cells directly, but sickle cell anemia patients have a distorted red blood cell shape. So the malaria parasite is not able to attack the red blood cell. So one benefit of sickle cell anemia is individuals with sickle cell anemia can live in areas with mosquitoes and whole lot of things because it becomes difficult for them to get affected by the malaria parasite. So the capital A, or capital H in capital H means a normal person, and this person is not a sickler. When we talk about AS and then HS, it means the person is a carrier. When you come to the genotype of the individual, the person is a carrier. But when you come to the phenotype of the individual, the person is normal. And then when you have SS, it means the person is a sickler. The phenotype and the genotype is showing sickler. Sickle cell anemia can be controlled when a carrier does not marry a carrier, or a sickler because there's a possibility of giving birth to a sickler. So the best way to avoid giving birth to a sickler is when a carrier of a sickle cell anemia and a carrier of sickle cell anemia prevents pregnancy, or the best way is when a sickle cell patient does not marry a carrier of a sickle cell. Genetics of blood. The ABO blood system. The genetics of blood group system says that the inheritance of blood group is determined by a single pair of genes, for which there are 3 alleles, the A, B, and O. Everybody inherits 2 genes from parents. When a gene is inherited, it produces its antigen on the surface of the red blood cell. So assuming you inherit A from your blood group A from your parents, an antigen is produced on the surface of the red blood cell. Individuals in the blood group A produce antibody B, whilst those in blood group B produce antibody A. This antibody B is a small letter B, and then the antibody A is a small letter A. Antibody A is against antibody small a is against antigen big A. Likewise, antibody small b is against antigen big B. Group O contains no antigens but has both antibodies A and then small b. Antigens are protein molecules found on the surface of a cell, like red blood cells. Antigens are the red blood cell on the red blood cell determine the blood type of an individual. So the antigen on your red blood cell, we first realize that antigens are produced on the surface of the red blood cell. So these antigens on the red blood cell determine the type of blood group you are. An antibody is a protein produced in the blood plasma due to the presence of antigens in the blood. Let me take it again. Antibody is a protein produced in the blood plasma due to the presence of antigens in the blood. They are produced by lymphocytes in the lymph nodes. They destroy microorganisms during their relation with the antigens. Blood transfusion. If a person's serum contains antibody B, he should be given blood from a person with blood group A. Group O people have no antigens, so their blood functions quite simply in other people. Group O people are therefore referred to as universal donors. Group O individuals can donate blood to Group A, Group B, Group AB, and Group O because they are universal donors. They don't have antigens in their blood, so it's easy to transfer their blood to other blood groups. People with blood group AB can receive blood from any other group because their serum contains no antibodies to fight against antigens. So since, like in the case of group O, they don't have antigens in their blood, and in the case of blood group AB, they don't contain antibodies to fight against antigens. So they are therefore referred to as universal recipients. They can take blood from any kind of blood group. They are very lucky. Okay, so we go on to resource factor. Resource system was first discovered in rhesus monkey. That's how we call it resource factor. Some people possess red blood cells containing an antigen called resource factor. Their blood is referred to as resource positive. Others lack resource antigens and are called resource negative. If the resource negative recipients sequentially receive doses of resource positive blood, the resource antibody will cause agglutination of donors' red blood cells, often with fatal results. So a person may have A positive, A negative, B positive, B negative, AB negative, AB positive, O positive, or O negative blood group. The positive and the negative means it's a resource negative, and then a resource positive, and A positive means it possesses resource factor. A negative means the blood group A possesses no resource factor. During blood transfusion, resource negative person can only receive blood from another resource negative person. So if you are A negative, you can only take blood from an individual who is A negative. A person with resource positive factor can, however, receive blood from a resource negative factor. Unlike the ABO system, rhesus negative blood does not automatically contain the rhesus antibodies. So this brings us to the end of this lecture, and these are our references. So thank you and have a good day. I hope all we discussed earlier is understood, and then I'll advise you to go over the slides, go over the video, follow everything like two times, 3 times, and then it will be really easy for you to understand and master genetics. You're also advised to watch more videos on YouTube concerning the the characteristics of genetics. Thank you.